Hi, we're releasing this video on the 20th anniversary of this paper from our research group, which was identified the first gene that implicates NAD metabolism in axon degeneration. And in the next 20 minutes or so, I'm going to give you a very quick run through some of the key findings of that paper and this research that is subsequently made possible in understanding the roles of this mechanism, which we now call programmed axon death, in disease, including human disease, and the drug discovery targets that are coming from that. And if you only have 30 seconds, the key take-home message of this introduction is that Wallerian degeneration is a well-understood and preventable, potentially fully preventable mechanism of axon death that is directly relevant to human disease. And it began actually in 1850, this story with Augustus Waller, who first saw what he called the coagulation and curdling of nerves distal to the site of an injury in the tongue of a frog. And he speculated at that time, and now we know he was basically correct, that this would be relevant, this injury-induced degeneration would be relevant to what he called nervous diseases. So these are, today we will say neurodegeneration in when there is no physical injury. And that became possible to test when a mutant strain of mouse came along in 1989, studied by Hugh Perry and Michael Brown at the University of Oxford. And those mice had acquired, although they looked and behaved exactly like wild-type mice, and they thought they were wild-type mice, they happened to have a spontaneously occurring genetic mutation that conferred a protective phenotype on their axons. And when they cut nerves in these mice, they found that the axons didn't degenerate at anything like the normal speed. In fact, they took 10 times longer than normal to degenerate. And legend has it that Hugh then called up Michael Brown, and his quote was, "Go cut the nerve again," because the most likely explanation at the start for this bizarre result was that the nerve had not been cut properly. But they did cut the nerves again. They went on to look at other mice, and they compared that breeding colony with other breeding colonies, and they found that every mouse in this breeding colony had this phenotype, but it was unique to this colony. And then they started looking at the genetic inheritance of this neuroprotective phenotype. They found it was a dominant phenotype, and they began teaming up with Mary Lyon just down the road at MLC Harwell to do the genetic mapping of this locus. In other words, where in the genome of those mice was the gene that was conferring this protective phenotype. And that's around about the time when I joined Hugh Perry's lab, and my job was to eventually identify the gene that was conferring this phenotype. That took about 7 years, but we got there, together with a huge amount of help from Laura Conforti and other colleagues. And it turns out the mutation in these mice was a very bizarre genomic rearrangement that had the effect of fusing together parts of two normal genes to make a completely novel gene in these mice that would not occur anywhere else naturally. The key business end of the predicted protein is an NAD synthesizing enzyme called Nmnat1. This synthesizes NAD from its precursor nicotinamide mononucleotide, or NMN. Remember NMN because it's going to come back later in this story. The end terminus of that protein was derived from a ubiquitin ligase called UB4B, and we now know from extensive structure function work from ourselves, Mark Freeman and Jeff Milbrandt, that the end terminus part of this protein is not required for the phenotype as long as that NMNAT activity is directed into the axon. And that turns out to be the key function of the end terminus, is to relocate the NMNAT activity that synthesizes NAD from the nucleus, where Nmnat1 normally is, into the axon. And we showed that this was indeed the causative gene by replicating that phenotype in transgenic mice, and that structure function work actually essentially told us that it is a gain of function mechanism for Nmnat activity in axons. A logical next question, though it took us the entire field a little while to realize that this was required, is if gain of function of this activity protects axons, does loss of function of this activity underlie the normal process of Wallerian degeneration? And that's work that we eventually did in our lab through John Gilley, And the answer indeed is yes, but there is a twist. So the protein involved in the chimeric protein, Nmnat1, is not the one that is missing from axons to cause Wallerian degeneration. That is a different isoform of Nmnat called Nmnat2. And when John knocked down and eventually knocked out that protein from cell cultures initially and in mice eventually, we found that you get either the degeneration of axons that are already formed or with constitutive knockout, the inability to grow long axons in the first place. So this is a lethal phenotype in the mice because they don't innervate, among other things, they don't innervate the diaphragm. So Nmnat2 is essentially the protein that normally prevents our axons from degenerating. So what is the process that it's actually preventing? Well, understanding of that comes from a very different direction of research, which initially was to replicate the mouse slow Wallerian degeneration phenotype in Drosophila. So Mark Freeman and colleagues were among the groups who first did this. They took the mouse cDNA, they expressed it in transgenic Drosophila, and they found the same phenotype. So this was clearly a very well conserved mechanism, right from invertebrates right through to mammals. And structure function analysis showed it was basically the same features of that cDNA that would confirm this protection. And Mark then said, well, this validates Drosophila as a platform to understand other genes involved in this process and set up his screen to identify the effector, in other words, a protein whose job it is to kill the axons. And that's where SARM1 comes from after screening about 10,000 different fly brains to look for individuals that were not shown the normal rate of axon degeneration. They eventually identified 3 loss of function mutations in the same gene, all of which protected injured axons. That gene is a Drosophila ortholog of SARM1, BSARM, and then together, and also with Mark Levine, we teamed up and looked at the SARM1 null mice, which were already existing at Cornell at the time, and found that these exactly replicate the slow Wallerian degeneration phenotype. So to formally prove that SARM1 was acting in the same pathway as Nmnats, it was necessary to ask whether removing SARM1 could block the degeneration induced in the most specific way by removing Nmnat2. So if you remember, the Nmnat2 null mice have this axon growth lethal phenotype, and remarkably that could be completely rescued by knocking out SARM1 as well. You see the mice here at 8 months of age. They clearly have full diaphragm innervation. Together with Richard Ribchester, John Gilly in the group showed that there was full rescue of the distal leg muscle innervation right through to old age in these animals. And this was clearly a full rescue of the phenotype. Now, this places SARM1 clearly downstream of Nmnat2, so that it's a loss of Nmnat2 that disrupts the repression of SARM1, some kind of activity of SARM1 exerted by Nmnat2 in this NAD sensitizing enzyme. And interestingly, Jeff Milbrandt and Aaron D'Antonio subsequently showed what that function of SARM1 is. They found it's an NAD degrading enzyme, so effectively the opposite of Nmnat2 in a simplistic kind of way, that degrades NAD, and somehow through that or another related activity, possibly, which are still beginning to emerge at the moment, initiates the process of axon degeneration. There is, of course, still the question of how does Nmnat2 or the lack of Nmnat2 talk to SARM1, and for that we focused on actually the substrate of Nmnat2, this Nmn that I mentioned earlier, because that in an injured nerve increases, accumulates in that injured nerve by approximately 2.5-fold before the axons actually start to fragment. We wondered if this might be a signal that was promoting axon death. And there's several lines of evidence for that, but one of the most compelling pieces of evidence is shown here. This was removal of Nmn or sequestration of Nmn by a mechanism different from WldS. So WldS would maintain the removal of Nmn when the NMNAT2 enzyme, which has a short half-life, has been lost, and WldS has a longer half-life. But we found a completely independent way of removing the Nmn by ectopically expressing a bacterial enzyme called Nmn deamidase. And that, as you can see here, very strongly protects injured axons. This phenotype is as strong as the original WldS mouse, as strong as the Salmon null mouse, and it's clear evidence that Nmn promotes axon degeneration. We didn't at that time know about Salmon NADase, it wasn't possible to actually directly show that an Nmn was inactivated, but that did come subsequently from the group of Zhongguan Zhao in Beijing, who that showed, and many others, including ourselves, have now replicated this, that Nmn increases the rate of NADase activity by Salmon. And this now appears to be how Salmon is regulated, because clearly it's very dangerous. If it's a protein designed to kill axons, you can't have it just going crazy in a normal healthy axon. It has to be repressed in some way. And it's the action of NMNAT2 in keeping the Nmn levels low, and also we now know keeping the NAD levels higher, is important for keeping Salmon normally at a basal activity. So the short story then is that Salmon kills axons when it is activated, but normally it's kept in an inactive or basal form by the action of NMNAT2 upstream, and the way it does that is by limiting the amount of Nmn in the axon and raising the amount of NAD. And when NMNAT2 is lost, they go in the opposite direction. Nmn goes up, NAD goes down, Salmon becomes more active. And in doing so, Salmon then degrades NAD and some other substrates, such as NADP, and it synthesizes other products from that reaction, which include a number of calcium mobilizers. Now, there isn't time here to go into the longer mechanism of this pathway, which we now call programmed axon death, because it's not only cutting axons, as Waller did, that can activate it, as you've seen already. It's the removal of NMNAT2, for example, and you will see other examples later in the talk about how you can activate this mechanism without physical injury. So programmed axon death. There are many other steps in this pathway, with some of the key authors that have discovered those listed there. There's actually also still an ongoing developing area, so there's a lot still to find out about this, about other events downstream of Salmon, other substrates of Salmon, other events upstream of NMNAT2, such as other ways of new ideas about how NMNAT2 becomes turned over and being a short-lived protein. And all of this, of course, have come from that discovery, directly or indirectly, of the initial gene that could block the pathway, that told us all that it was possible to genetically alter the outcome of this mechanism, and also that NAD biology was somehow very much involved in that. So that's what we currently know about mechanism. What about the roles of programmed axon death in disease? Well, there's a number of questions, a number of ways to approach that question. It's been known for decades that the morphology of axons undergoing degeneration in disorders such as peripheral neuropathies, ALS, multiple sclerosis, resembles that of the morphology of an injured axon undergoing degeneration. But that morphology similarity doesn't necessarily mean shared mechanism. So can we use the ability now to interfere with the rate of degeneration after an injury-induced lesion and ask whether similar mechanisms can be involved in the non-injury disorders and therefore delayed by the same genetic mutations, the overexpression of NMNAT or the removal of Salmon. So can elevating NMNATs or removing Salmon alleviate disease? But conversely, can lowering NMNAT2 or activating Salmon cause disease? And you've already seen a hint of that, because you've seen that completely removing NMNAT2 actually causes an extremely severe, what you might call a disease, so the developmental disorder, the inability to grow long axons. And then, crucially, not just in animal models, but in which human diseases does this happen, and how can we tackle that? And ultimately, can drugs be developed to block this mechanism? So the question of whether blocking the pathway can alleviate disease is best addressed in disease models, animal models and cell culture, where we can overexpress NMT, as in WldS, or remove SARM1. And there are, of course, genetic and toxic models of disease, and both of those are shown here. There is two models both of which disrupt microtubules and therefore axonal transport. There is a genetic disruption of putting together the full complement of microtubules in the PMN mice, progressive motor neuronopathy, and there is a toxic disruption of microtubules by the cancer chemotherapeutic vincristine. And as you can see, in both cases, the presence of WldS rescues or temporarily rescues those axons. And the key point here, as well as the role in disease, is you do not need physical injury to activate this degeneration mechanism that proceeds through WldS. Many of these disease models have also been shown to be alleviated now by SARM1 removal as well, especially the vincristine model. There are other models too, such as glaucoma, here a rise of intraocular pressure, again generating a WldS-sensitive degeneration mechanism. So that was the prevention or the alleviation of disease by preventing the mechanism. What about causation of the disease by activating the mechanism? And as you've seen already, one way to do that is to remove NMT2. So other human diseases involving a deficiency of NMT2. Well, sadly, the answer to that is yes. There are rare forms of polyneuropathies where NMT2 is biallelically mutated, and we have worked together with clinical colleagues and Giuseppe Orsamando in Ancona to show that these are loss of function variants of NMT2. In the most severe case, this results in a stillbirth phenotype, so resembling the null mice, but possibly even more severe because these individuals are prematurely terminated and don't ever even develop skeletal muscle or proper brain structures. And those were very severe full loss of function compound heterozygous variants of NMT2. There are also more modest loss of function of variants for NMT2, and these results, first of all, in mice, if we take down the NMT2 expression level, not removing it completely, but take it down to very low levels, we find an early onset sensory phenotype and a late onset motor phenotype, both of them subclinical, not severely impairing the life of the animal, but they are definitely detectable in neurological tests. And again, there is a similar or related phenotype in humans with a partial loss of function, biallelic loss of function of NMT2. And this manifests really as erythromelalgia, burning pain in the distal limbs, but it's also associated with polyneuropathy, so loss of motor and sensory and possibly some central axons affecting long axons in different parts of the nervous system. So if removal of the protein that inhibits this pathway, NMT2, can activate the pathway in disease, can also the activation or the increase of SARM1, the pro-degenerative protein, the effector of this pathway. And there's evidence of this so far in ALS, in the Project Mine whole genome sequence database of sporadic ALS patients and controls. There is a patient-specific cluster of missense and microdeletion variants in the inhibitory arm domain, the N-terminal domain of SARM1. Now, disruption of the inhibitory domain, you would expect to be gain of function that might therefore be a harmful event in those patients. And we've, first of all, we've tested the enzyme activity of all of those variants, and approximately half of them do indeed show a massive increase in the basal activity of NADAs for SARM1, the grey bars here. You can see there's actually a 20-fold increase over the wild-type level, which is higher even than the NMN-induced wild-type protein, which itself should normally be enough to kill axons. So there are very interesting questions here about why this disease in these patients is not even earlier onset. Is there some kind of compensatory mechanism, which is a very important question to take forward. And are these actually harmful variants? Well, yes, that is the case, at least in this cell culture model here. When we express them in mouse neurons in culture, then we see the death of the cells that are injected with a mutant version of the protein, and these can be at least rescued by nicotinamide riboside, the NAD precursor, although they still remain somewhat fragile if they're exposed to any other kind of stress, even in the presence of NR. So we've had activation of the pathway by removing NMT2, activation of the pathway by disrupting the autoinhibition of SOM1 through genetic mutation. What about environmental activators, exogenous activators? So NMN is already known to be the natural activator of SOM1, or a natural activator within normal axons or injured axons. But there are analogues of NMN that we now know can do the same job. And we came across this through studying this chemical called Vacor. It's a nicotinamide analog, which is a disused rat poison. It was banned because it had occasionally been humans exposed to it, who very many of those developed a polyneuropathy phenotype subsequently. So we were interested to know, could this cause a degeneration that was SOM1 dependent? As you see here, very strikingly, yes, absolutely, the effect is SOM1 dependent, because it immediately causes death of both the axons and the neuronal cell bodies in the cell culture here. But the SOM1 null neurons are completely resistant to Vacor. And this was the case no matter how many times we kept adding Vacor into that cell culture medium, that we never saw any death for those axons. We also replicated this in vivo in a model in mouse retina, and there is very strong rescue of those Vacor-induced neurons and axons by the absence of SOM1. And we now know through structural work together with Boston Cobra that Vacor's downstream metabolite VMN, Vacor mononucleotide, and the analog of NMN, also binds to the arm domain and very potently activates SOM1. And that raises many important questions about what other activators of SOM1 there may be in our environments, and potentially other pesticides and other related chemicals, and that's one direction that we are now taking the research. But in summary, so far, we've discussed at least 5 ways to activate SOM1. So we started off with axon injury, which depletes the amount of NMT2 in axons because it's a short-lived protein and they get steadily degraded. We talked about genetic disruption of NMT2 in the mice and in polyneuropathy cases. We've talked about genetic hyperactivation of SOM1, which is associated with sporadic ALS. We've talked about the impairment of axonal transport with toxins such as vincristine and paclitaxel, and now we've talked about compounds such as Vacor that can be metabolized into direct activators of SOM1, and there could be any number of other mechanisms through which SOM1 is activated. There's a lot to still to find out about this pathway. But thankfully, it's not all bad news. There are a number of drug companies trying to target SOM1 or other steps in this pathway to alleviate neurological disorders through blocking programmed axon death. And here are some, actually, at least 5 different ways that are known in animal or cell culture models to be able to block SOM1, many of which are being followed up as potential therapeutic avenues. One of these is antisense oligonucleotides that take down the expression level of SOM1, and we have a paper coming about that showing effective rescue in vitro models from vincristine and other activators of the pathway. There are precursors of NAD that are undergoing clinical trials as a way to impair the pathway. We've already shown that sequestering NMN can do so, but perhaps the most popular approach that is being taken, of course, is inhibition, direct inhibition of the NADA's activity by understanding the structure and finding inhibitors that can interfere with that enzyme activity, but also interfering with the regulation of SOM1. There's many different ways to target this pathway, and they are many or all of them are being followed currently. So in summary then, NMT2 and SOM1 have opposing effects on axon survival and on the level of NAD in those axons, although whether NAD itself or some other related metabolites or the product of one of those reactions is the actual direct cause of degeneration, I think that's still work in progress. That's still a very important thing we need to find out. And in human disease, biallelic loss of function mutation of NMT2 causes rare polyneuropathies of varying degrees of severity. Gain of function mutations hyperactivating SARM1 appear likely to contribute to ALS. They're certainly enriched to some degree in ALS, although they are very rare mutations, even in ALS cohorts, so it's actually very difficult to get to the sufficient power to get genome-wide significance, but they are clearly harmful mutations, and they are present in ALS patients more than they are in the general population. And both effects, both the absence of NMT2 from axons and the direct activation of SOM1 can be mimicked by neurotoxins, vincristine or Vacor, 3-asdalpyridine is another paper just out, preprint just out from the D'Antonio group, and there's a number of different ways in which SARM1 and NMT2 can be impacted by exogenous toxins. And the pathway can be completely blocked. We know that from the full rescue of the NMT2 null animals by removing SOM1, and actually also the very strong rescue, the Vacor-treated neurites, also strongly suggests that there's a full block in that case as well. So if you activate the pathway specifically enough, you can completely prevent it by removing SARM1, and it is a druggable pathway. There are many more questions to answer, potentially over the next 20 years. Other human diseases, that is a key question we need to answer. Which other human diseases may involve this pathway? Certainly the animal models would suggest a wide possibility of other disorders to investigate: glaucoma, multiple sclerosis, diabetic neuropathy, and Parkinson's disease are some of the most interesting possibilities, all of which have been implicated in the animal models, in the rescue effects in animal models of those disorders. We need to find ways to identify specific patients where it contributes, who have got the loss of function NMT2 or the gain of function SARM1 variants. We need to work out which of those potentially five or more therapeutic angles are the most effective, or which combination. What other environmental toxins might impact on the pathway. There's a very important basic science question about where do NMT2 deficient axons, or the ones rescued by removing also SARM1, where do they get NAD from? We don't know actually for sure that any other NMT enzymes are present in axons. It's not clear that NMT3, what is reported to be the mitochondrial isoform, but only present at very, very low levels in brain, it's not clear at all that that protein is in axons. So there's very important questions, perhaps some very important things we don't yet understand about axonal NAD metabolism. How does NMT2 as a short-lived protein get to the end of our long axons? Is there a role for local protein synthesis there? There's a lot of questions still to answer in this pathway, and I guess a lot more surprises down the road. So thank you for listening. We'd love to hear from you. We'd love to start more regular discussion online through Twitter or privately through email. And of course at conferences, when we can go to conferences again. And do leave comments on our preprints. We really want dialogue in this area, and look forward to more interest and discussion about this really fascinating mechanism. Thank you.